everybody. Welcome to The Mixer. I'm your host, Andy Marshall, here again with my buddy, Juan Carlos Lopez.
Hello, Andy. Good to be here.
So, JC, who's our guest today?
Andy, today we have a very exciting episode of The Mixer. We have Kiran Musunuru, who is a professor of medicine at the Perelman School of Medicine at the University of Pennsylvania. As you know, Kiran has been a pioneer in the study of genome editing, and he has been instrumental in bringing base editing towards clinical application. He recently co-organized a Keystone meeting on Precision Genome Engineering, and he's going to share with us some of the insights that transpired at the meeting.
That sounds great. We should probably also mention he's a scientific co-founder and advisor of Verve Therapeutics, which is a biotech that's pioneering some of the base editing therapies that we're going to be hearing about. So let's get going.
Let's go.
Welcome, Kiran. It's great to have you here on The Mixer. So we wanted to talk to you because you recently co-organized a Keystone, which was called Precision Genome Engineering. So it'd be interesting to just for our audience just to hear a little bit about the meeting itself, like what was its genesis, whether it's part of a series, and some of the aims of the you and the other co-organizers that you had for the meeting, and then some of the key themes perhaps that emerged.
Absolutely. Well, first, let me thank you, Andy, and Juan Carlos for inviting me to be on the Mixer. And let me get started. So the Keystone Symposium on Precision Genome Engineering, it's one of a series, as you suggested. It's been going on for quite a few years now. So it's an annual event. Not all Keystone Symposia are annual, but this is one of the more popular ones, one of the very well-attended ones. And it has become an annual fixture. And the other distinctive feature of this particular one is that often Keystone meetings are paired up with other Keystone meetings, joint sessions. The idea being to, if there overlap or some commonality between two themes, have them occur at the same place, same time, and then you can have some joint keynote sort of sessions, but you'll also have the separate sessions. And so it does foster some intermixing of the communities. And so this year, it was actually paired with basically mRNA delivery. Or RNA delivery, or I should say nucleic acid delivery, so more broad than just RNA, mRNA. And that actually, if anything, ended up being a little too close in the sense that I found myself really wishing, wow, I'm really enjoying what I'm seeing here in the Precision Genome Engineering room. But wow, at the same time in the other room, just right next door, these really cool talks on delivery that I would really like to see, just because genome editing and delivery are really so intertwined. So I felt like, wow, it's kind of too bad that these two meetings were together in the sense that, you know, in a way, I would have loved to have gone to either conference at different times and to have it all in one place. For me, at least being the organizer of one, I was basically stuck with one. And so missed out on a lot of what I'm sure were very interesting talks. I know they were interesting because other people who were not so, didn't have the fidelity I had to have to genome engineering, you know, got to go back and forth between the rooms. And so I got to hear, you know, sort of secondhand what was going on in the other rooms. Like, wow, I wish I could have been in there, but sadly I could not. So to answer your question, you know, like some of the exciting stuff that went on, at least on my side of the resort, so to speak, and precision genome engineering. What I think this meeting emphasized in a very powerful way is just how much progress is happening on all fronts. So I can't say there is one single astonishing new discovery that just kind of blew everything up, right? That, you know, like just upended the field. That was not this kind of meeting. There wasn't like an announcement in the sense that, wow, we have like a whole new editing modality along the lines of, say, a base editing or prime editing. What we saw was a lot of elaboration and a lot of progress in the editing modalities that already exist, both on the technological side, but perhaps more importantly on the clinical side, clinical translation, actually starting to see some of these technologies actually make it to the clinic, actually have an impact on the patients who have been enrolled in the clinical trials. And of course, we're all aware of Exa-cel, the sickle cell and beta thalassemia treatment, getting approval from the FDA not that long ago. And then even for those technologies that are even newer and haven't even had the chance because they haven't been around that long to make it all the way down the clinical development path, you can see they're going to get there. you can see that progress is being made in preclinical models, in mouse models, in non-human primate models. And then it's only a matter of time before they get into the clinic and human patients start to receive doses of these, various types of gene editing technologies. And it's going to greatly open up the horizon in terms of diseases that can be treated with editing.
Maybe we could take this kind of one step at a time. If we're thinking about therapeutic modalities in terms of gene editing, Kiran, so obviously, there's CRISPR Classic, Cas9, and then since 2016, we've had base editing technologies kind of come online, and we're going to get into that, I think, a little more in terms of some of those are now already moving into the clinic.
Yep.
And we have prime editing, which has a lot of flexibility in terms of the types of changes that you can introduce, opening up this opportunity to actually do kind of knock-ins in a manner that's perhaps less chaotic than Cas9.
Yeah.
It's really powerful technology. But we've also started hearing about, serine integrases. People are talking about transposases. Even people have been talking about gene writing and retrotransposons.
Yep.
So could you fill us in a little bit about what you've seen there and what you think is exciting?
Absolutely. I can go through those one by one if that works for you. There's a lot to say on each of those ones that you mentioned. So let's start with CRISPR 1.0, as a lot of people think of it, standard nuclease editing. I think what was really exciting to see on the clinical side, there was a nice presentation by Intellia Therapeutics showing two different drug products, one intended for transthyretin amyloidosis, the second intended for an entirely different disease, hereditary angioedema. So two very different diseases, their clinical presentations, their pathophysiology, everything about them is different. They have nothing to do with each other, except for the fact that in each case, if you inactivate a gene in the liver, you can have a therapeutic benefit. And so what they showed very powerfully, and we already knew about transthyretin amyloidosis, the original paper came out in the summer of 2021. So, you're already talking, 2.5 years ago, showed great impact in terms of reducing transthyretin, the toxic protein levels in the blood in the first patients who were dosed. And they're, all the way into phase 2 at this point, treated dozens and dozens, maybe at least 70, maybe even up to 100 patients by this point. So that's been going well. So that's not news in and of itself. What's newer is the fact that they're now starting to discuss data from the second indication, from hereditary angioedema. And so what was nice to see in the talk was the juxtaposition of the two. And that's very powerful because what it really emphasizes is the programmability of the technology. What they were able to do is take their one drug for transthyretin amyloidosis and take that, essentially the same exact drug, and change just the guide RNA. Everything else is the same. The Cas9 nuclease is the same. The lipid excipients of the lipid nanoparticles is the same. Everything is the same except that guide RNA. And now all of a sudden you have a different drug product that works for hereditary angioedema. And so you have a series of patients who are treated with this second drug, patients with hereditary angioedema. And what was really exciting to see, and it's not absolute new data that was unveiled at Keystone. It's been out for a few months, and the New England Journal of Medicine paper came out actually right after the Keystone meeting. But showing the cohort of patients, the first cohort of patients who got this drug had an amazing clinical response. Before that, they were having pretty frequent angioedema attacks, which it's not necessarily fatal or anything along those lines, but it's very detrimental to quality of life. And in almost all of the patients, no more attacks in the few months after they'd gotten treated. And so I would say this is actually the first demonstration of an in vivo gene editing therapy actually demonstrably improving the quality of life of patients. Transthyretin amyloidosis, that was a great success, but that was all based on the biomarker seeing toxic protein levels in the blood go down. But it's harder to measure clinical outcomes. That takes a lot longer. That takes years. You have to do, randomized controlled trials to really be sure what's going on. Whereas with hereditary angioedema, you really could see the impact in the sense that these patients are just having much better lives. And this sort of parallels what we saw on the ex vivo side with Exa-cel, with the sickle cell / beta thalassemia drug, where those patients were having sickle cell crises. They were having, needing transfusions, having, a lot of pain, episodes of pain that required treatment, hospitalizations, and so forth. And most of those patients have not had any such events since they got treated. So now you have what is clearly and in vivo success, really improving patients' lives to go along with the ex vivo success. And to me, that's awesome, right? Like we're now really ripping and roaring right out of the gates because, it's ex vivo, in vivo. It's like now you have the full spectrum of disease.
That's a really key advance, yeah, to show clinical outcomes with an in vivo editing therapy.
Absolutely. Absolutely. That's a real advance to show that there are clinical improvements. Like this is not just a drug that's changing a biomarker or this or that. It is actually impacting patients' lives. Like there's no ambiguity about it. But then the other message that I think in a way is, to me at least, more exciting and has broader implications, as I said, is the programmability. Because now you've done it for disease 1, it seems to be working. You've done it for disease 2, it's definitely working. And, it doesn't take much imagination to say, hey, let's just tweak that guide RNA and target a third gene, an activated gene, and then you can treat some other disease. And so we'll see that happening. We'll see that happen with Intelli. I'm sure we'll see with other companies that are largely focused on nucleases. CRISPR Therapeutics, Editas, Metagenomi, which just went public. They're all sort of in this nuclease category. Now, the limitation there is that there are only so many genes to inactivate, particularly in an organ like the liver. There are only a handful of diseases, right? The vast majority of genetic diseases or even acquired diseases, that strategy is not going to work. And so then you have to go to the next generation CRISPR technologies, right?
This issue of programmability you alluded to, I agree with you that it's super interesting and a real advance. Now, the question is, from the point of view of clinical development, has regulatory science caught up with this programmability, right? Because you could say the way it sounds is that you could essentially swap the guide RNA and have a new drug, but the regulatory science will still require you to do all the safety and toxicology studies, everything that you need to do before you can go into the clinic, right? And in the gene therapy field, as you know, the NIH has this initiative called PAVE, in which they are trying to precisely avoid this. Let's keep everything the same and let's just put a different payload in the virus. And that way we can save ourselves a lot of preclinical development when projects get to the clinic. So are the regulatory agencies thinking about it in the same way?
Yeah. You hit the nail right on the head, Juan Carlos, with that question. It's very much on my mind. So by way of disclosure, as an academic investigator at the University of Pennsylvania, I'm the recipient of funding from the NIH from their Somatic Cell Genome Editing Consortium, which is intended to fund investigators like myself, largely on the academic side, to take in vivo genome editing therapies, this is restricted to in vivo, to the clinic, through IND-enabling studies to the clinic. And so one of the programs on which I'm working, and it does tie to the Keystone because a couple of my students presented on this work, so it's technically included in the Keystone update, if you will. But something we've been working on is the disease PKU, phenylketonuria, a very well-known genetic disease. Patients who manifest disease, it's picked up on newborn screening. It's actually the reason newborn screening was instituted in the 1960s. It was for this disease and the recognition that you start to incur neurological damage from day one if you don't actually try to manage the disease. The disease is caused by very high phenylalanine levels in the blood because the enzyme that breaks it down is defective, and if it builds up high enough, it's neurotoxic. And you can somewhat control it. Not totally control, but somewhat control if you have a strict low-protein diet. There are a few medications. They don't necessarily work that well, or they're challenging to take, or they're expensive, like daily injections. So there's still a great amount of unmet need in this disease. And so something we've been working on is developing base editing therapies to directly correct disease-causing mutations. Now, the challenge with a disease like PKU and most genetic disorders is that there are a lot of different mutations, or more properly speaking, a lot of different variants, genetic variants in the responsible gene that can cause a disease, right? It's not one size fits all. In contrast to sickle cell, where there's one variant that's responsible for all cases of disease. So you can have one size fits all solution there. And so what we've been doing is asking the question, okay, we can make different therapies for different variants. They have similarities. And this gets to your question, Juan Carlos. In some cases, you're using the same exact editor, whether it's a base editor as we're doing, or whether it's a prime editor or a nuclease editor or XYZ, and you're only changing the guide RNA. And so you have these two drugs, say, for two different variants in two different parts of the gene, but it's the same disease. And the editing works in the same way. It's just the guide RNA is different. The variant that's being fixed is different. But the fundamental mechanism and all the characteristics of the drug are otherwise the same. And so one advantage of my being on the academic side is I don't have to have the same sensitivities about my interactions with the FDA that companies do, right? For a company, it's proprietary, it's a trade secret, like you'll never get them to fess up to exactly what went on in a discussion with the FDA. As an academic, I couldn't care less about that. I'm actually happy to share with the world exactly what I learned from the FDA. And so I actually had a meeting with the FDA a couple weeks ago, not that long ago. And one of the questions I posed was exactly this question. We have these two drugs for PKU that are very similar. Essentially, like just changing the guide RNA, tweaking the messenger RNA a little bit because it's not exactly the same base editor. It's a little bit different, but it's largely the same. At the mRNA level, 99.5% identical. And we asked the FDA this question: can we streamline the regulatory process? Can we include these two drugs in the same IND application to allow us to start clinical trial? Or are these going to be treated like two different drugs and we have to do the full suite of IND enabling studies, whether mouse studies and monkey studies and so forth for these two drugs? And we could tell that the FDA is still very much thinking about this, right? They don't have definitive answers for any of this. And we're probably one of the first, probably not the first, but probably one of the first to approach them with this kind of question, even though it's kind of in the air. And the response they gave back to us was that if the editor is not the same, so even if it's slightly different, that it's going to be treated as two different drugs. They cannot be part of the same IND. However, there may be situations where if it's only the guide RNA that's different, it can be included in the same IND. Now that's very high level blanket advice. The devil's in the details, and it's hard to know, what are the circumstances where it would be allowable? What are the exceptions? I think we're going to have to figure that out, and the FDA is going to have to figure that out as we go forward. But there is some openness to the concept of programmability. It's just not totally clear what shape that's going to take, what sort of evidence the FDA is going to need from us, as the investigators, and from companies as well, to convince them that it makes sense to take advantage of this programmability, because we understand from what studies have been done that the drugs, it truly is the same drug. Its biodistribution is the same. Its toxicology is the same. Just changing the guide RNA doesn't change any of the fundamental characteristics. It's going to be just as safe regardless of which guide RNA you use. So go forward with it. So that's kind of where we stand right now.
I guess, Kiran, a corollary to that is the biology also needs to match between the case that you're using and then extending it out, because, for instance, if you're going after a different cell type, there's all kinds of different variables here that have to be taken into account. So it's not only the modality itself, it's the biology, pathomechanism behind the disease, how we understand that all of these things are going to come into that calculation,
They will, right? So, one interesting question is, we presented the simple case of, they're both for PKU. It's the same disease. It's the same organ we're targeting, the liver, right? You're right. That simplifies things. If we had gone to them and asked, we have two drugs that are exactly the same, guide RNA is different, but it's two different diseases. One is PKU and one is another metabolic disease, galactosemia. Is that permissible to include in the same IND? And so we're going to be approaching the FDA over the coming years with exactly that same question, because our ambitions go far beyond PKU. We want to tackle a whole variety of diseases. As I understand it, Intellia was not in a position to ask these questions of the FDA with its programs because one was so far ahead of the other, and they basically did the transthyretin amyloidosis program, and it had already gone to the clinic and was having success by the time their second one was ready. But it's the same sort of situation. You have the same drug, you're just changing the guide RNA, same target organ, the liver, but it is two different diseases. So what it might look like to have them in the same IND, to have them part of, say, an umbrella clinical trial, who knows, right? It's the Wild West, but it's going to be very fun to try to figure this out. A fun challenge, let me put it that way. It won't be simple, but I think it'll be a very interesting space to watch.
Yeah.
But anyway, we got sidetracked.
Yeah, I was going to say we can go back to the other modalities we're discussing.
Yeah. Well, I already sort of got into base editing by virtue of my example of PKU, but other things that are going on, and in the same way that I did for nuclease, let's talk about the clinical successes that we're starting to see with base editing. So I can talk about two, and they were both presented at the Keystone. One by Wasim Qasim from University College London. So he gave a nice talk on the work he's been doing ex vivo therapy. So now you're talking about outside the body, taking cells, in this case, T cells outside the body, and then treating them, in this case, with base editors to effectively create cancer immunotherapy. And was able to get this to work very well. And if you've been at all paying attention to the field, you've heard of Alyssa, the young girl who had refractory leukemia, had gone through all her treatment options. There was nothing left. And his team was able to craft this new therapy using base editors, which at the time was only 3 or 4 years old, using that to engineer these cells, knock out different genes to engineer this therapy, this immunotherapy, CAR T, and then administer it to Alyssa. And it worked. And she's in a remission. And she's healthy to all appearances. An incredible success, especially coming not that long after the success with nucleases ex vivo deployed for sickle cell and beta thalassemia. And so, it's great to hear him give updates on that work and his ambitions going forward. The other talk, again, by way of disclosure, it was a talk I gave by virtue of my involvement with a company called Verve Therapeutics. So my disclosure is I'm a co-founder of Verve Therapeutics and continued to be a scientific advisor to the company. I don't speak for the company, and I did not speak for the company. I have to be very clear about that. I was not representing the company at the Keystone, but I did summarize some of their publicly presented data. And the real excitement there is the first in vivo base editing therapy administered to patients with familial hypercholesterolemia, a genetic condition that causes them to very high blood cholesterol levels, so high that they have very bad heart disease. So these are really, really sick patients. And 10 patients have been enrolled in the phase 1 clinical trial with a base editing therapy delivered by lipid nanoparticles in the same way as Intellia's therapies. So using messenger RNA, encoding a base editor, using a guide RNA, in this case, targeting the gene PCSK9, which is a cholesterol-regulating gene, with the purpose of inactivating it. And so it's kind of like a nuclease in that the goal is to turn it off. The difference is that here you're only changing one base, one letter, out of the 3 billion letters in the genome. But you're doing it in exactly the right place to turn off the PCSK9 gene. So the effect is the same. You're turning off a gene, but it's in a cleaner, more precise way. And this was administered to the first 10 patients at varying doses. It was a dose escalation study, single ascending dose, where you start with a very, very, very low dose, almost a homeopathic dose, just to make sure it's safe. Do that in a few patients, go up to the next dose, do a few patients, and so forth. And what was evident just from the first 10 patients is that there is a dose-dependent effect. The higher the dose, the more reduction of LDL cholesterol levels, the so-called bad cholesterol levels in the blood. And the patient so far who has received the highest dose had a 55% reduction of LDL cholesterol levels, fell by more than half as a result of getting this therapy. And what's remarkable, not surprising at this point, because Intellia has shown data like this as well, but what's remarkable is that in the data that's been presented publicly so far by Verve Therapeutics, that reduction in LDL cholesterol has been rock steady stable for more than 6 months so far. Like it comes down and then it does not budge. It is down 55% and it just stays there. So we're at 6 months as of the latest data release. My expectation, my prediction is it's going to be there for the lifetime of that individual. So you can see the potential here. Wow, okay, so base editing, it's now been deployed in human beings. It is working. And so that's great. As a cardiologist, because I am a cardiologist, that's that makes me very happy because now we have this powerful new tool to tackle heart disease, which is the leading cause of death worldwide. But again, there's a bigger picture. Just like with Intellia and transthyretin amyloidosis and hereditary angioedema, there's a bigger picture, which is that now that we know base editing works in human beings, and base editing has the unique advantage over nuclease editing in that it can make precise changes. We can do much more than just turn off genes. With Alyssa, with CAR T, it was about turning off genes. In the hypercholesterolemia trial from Verve, with PCSK9, it was about turning off the gene. But very, very soon, what we're going to see is base editing being used to correct disease-causing variants in the body. And that will open up treatments for a whole variety of rare genetic diseases, where the key is not to turn off a gene, because that's not going to do anything for you, but it's to actually fix, correct, repair, whatever word you want to use, the gene, restore the function of a faulty enzyme or some protein that has an important function that's somehow driving the disease, and then effectively be able to, we don't use the word cure lightly, but potentially cure diseases, and potentially do it at a very young age. So we've already talked about PKU. That's one such disease where, as I mentioned, we've been able to show, at least in mouse models, that we can correct 2 different PKU variants in different parts of the gene with base editing, correct that variant. And what we find is when we do this with lipid nanoparticles, with messenger RNA encoding a base editor, with guide RNA, within 48 hours of treatment, the disease is entirely resolved. Those phenylalanine levels that started super, super high are now down to normal. 48 hours, that's all it took. Entirely corrected the disease. Stunning. And just imagine doing that in a human being. And so hopefully my academic group, with the funding we have from the NIH Somatic Cell Genome Editing Consortium, in a few years, will complete the IND-enabling studies and get permission from the FDA to actually take this into human beings.
Kiran, this is a really fascinating point that you're making, and really speaks to the transformational aspect of base editing. But the other thing that maybe we should kind of refer back to Wasim Kazim's work is the point about the safety, the increased safety of base editing. Yes. So obviously with traditional Cas9, you're creating these double-strand breaks in the DNA. And if you're creating multiple edits, then you kind of get this problem of unwanted recombination in the chromosomes that you really want to avoid. So can you just speak to that other interesting advantage that comes along with base editors?
Yeah, so the potential for multiplexing, I think, is what you're really getting at. The idea that you can mix several guide RNAs with an editor, and that editor will now go to several places in the genome and make the desired change. If it's a nuclease, that means double-strand breaks. And so you're going to get multiple double-strand breaks throughout the genome. And anytime you have that scenario where there are multiple breaks at the same time, there's the potential for what I think of as genomic mischief, pieces of chromosomes coming back together in the wrong ways, which may be okay, right? It all depends on context. So if you are in a highly differentiated quiescent cell type like neurons, like hepatocytes, the whole genome's there. It's just not strung together in exactly the right way, but it's probably not going to have a big functional effect. That's not a given. If you happen to hit an oncogene, if you happen to hit a tumor suppressor gene, maybe there are negative consequences, but you have to get pretty unlucky for that to happen. If you're in a stem cell, like say a hematopoietic stem cell, it's kind of a different scenario, because if you have a reshuffled genome and that cell has to give rise to a whole niche of cells, like say the entire hematopoietic system through many, many cell divisions, even with the first division of that stem cell that has an abnormal genome, you're not going to get normal genomes segregating into the descendant cells, right? You're going to get imbalances, you're going to get aneuploidy and so forth. And so there's the potential for carcinogenesis arising from that, right? So context is important. I hate to, paint with too broad a brush and say nuclease bad, base editing good, because it's not nearly as simple as that. But you're absolutely right. Like base editing and prime editing, and, those are the 2 precision editing technologies. They have the advantage that you're not, at least not directly making double strand breaks. You can multiplex and get single letter changes in, say, several different places in the genome. And what's happening in one place doesn't affect the other. Or the way I like to think of it is, what happens in Vegas stays in Vegas. Like what happens on chromosome 1 stays on chromosome 1. What happens on chromosome 6 stays on chromosome 6, right? They don't interact with each other in any way when you're using something like base editing or prime editing. And so we've seen that principle applied very nicely by Wasim in the treatments that he's making for patients like Alyssa.
So one other point that I'd really like you to kind of elaborate on for our audience is, I think with the PCSK9 story, there's this argument that's being made that this will kind of lay down a marker for base editing being useful for broadly available conditions rather than these kind of very rare conditions. And this brings in the practicalities of manufacture, distribution, pricing, all of these issues that tend to, if you're thinking about ex vivo gene-edited cell therapy, it's really difficult to think of that as something that will be broadly available to many, many people. Can you talk a little bit about the elements of the base editing platform for PCSK9 that you think are going to allow it to be more broad?
Yeah, so I mean, it really comes down to a few things, right? And you can really tie it to the components, right? There's the basic delivery vehicle, and by and large, we're talking about lipid nanoparticles. There are other ones in play, right? There's the more traditional adeno-associated vector AAV delivery approach, and that's fine. For some organs, you don't have a choice. That's the only way to deliver there. If you're talking about the liver, lipid nanoparticles really seem to be the best option, at least for now. There are virus-like particles of various types that are, being tested. We actually saw some presentations of that. I'm more on the delivery side of this Keystone symposium. I didn't necessarily get to see all those talks, but as I said, but a lot of energy there in using virus-like particles and attaching different doodads, so to speak, antibody fragments or chemical modifications to get them, lipid nanoparticles or virus-like particles to go to different places in the body. So that's one very important element, is like, where are you directing your therapy? But once you've gotten it there, and that's a big assumption that you can, but let's assume you can get it to where you want to go, then the editing machinery actually becomes repurposable or programmable, as I said before, right? So you have your messenger RNA that encodes the base editor. And the base editors come in different flavors, but it's pretty straightforward to just test a bunch of different variations and then figure out which one is best matched to the variant you're trying to correct or the gene you're trying to inactivate. And then you have the guide RNA, right? So you have your lipid nanoparticle or your virus-like particle or whatnot, then you have the base editor, and you have the guide RNA. So it simplifies down to a pretty, reasonably handled mix of things. There aren't that many variables you have to worry about. In contrast to prime editing, which we can talk about in a moment, which is more complicated, but at the same time more versatile. But what's really remarkable to me, and again, this really came out of Keystone. There was a whole session, a workshop where there were half a dozen talks from different groups that are all using base editing to go after different diseases. We already talked about CAR T for leukemia. We talked about hypercholesterolemia. We talked about PKU. But, we saw spinal muscular atrophy in one of the talks. We saw thoracic aortic disease in another talk. We saw a rare cardiovascular disorder called pseudoxanthoma elasticum in another talk. And so on and so forth. And you can see, wow, the space editing technology, at least at the preclinical stage, now that the tools are there and there's a clear path to the clinic, it's been now proven out in human beings, seeing a lot of energy from a lot of groups around the world saying, okay, well, let's take the disease where we think we can make the most impact. Let's take these tools. It's not too hard to figure out how to get base editing to do what you want it to do, within its technological constraints. And you can quickly, rapidly, demonstrate its efficacy in mouse models, and then you can kind of take it from there. And so I think to your fundamental question, which is about cost and access, I think a lot of the accessibility is going to be tied to streamlining. Can you actually streamline the process of discovery? Can you make it very, very straightforward to say, okay, I have a variant in gene X, and I need to target an organ X, and are there enough, well-validated pieces lying around that I can just kind of, switch out whatever I need, take from this toolbox, take from this toolbox, and within, ideally, a matter of weeks, and if not weeks, at least a few months, hopefully, be able to come up with an optimal solution for that variant, that disease, and that target organ, make your drug, and then be able to give it to the patient. Now, of course, that brings in the second piece, what Juan Carlos referred to earlier, which is the regulatory piece. You need to work with the FDA and come up with a streamlining so you don't have to take that new drug and spend, 3 years and $10 million to prove that one flavor of a base editing drug, but instead be able to rely on precedents and say, well, it's already been done, half a dozen times across all these different diseases. We understand the drug itself. We understand its safety profile. We can be satisfied that it's safe to go forward and then get an expedited regulatory process. And so I think if you cut out a lot of the work involved in discovery and you cut out a lot of the work that's entailed in regulatory approval, that's what we have to do to be able to get this down to an affordable timescale and affordable price and be able to greatly increase accessibility to a large number of patients. And I'm not just talking about a one-size-fits-all drug like sickle cell, which is where most people go when they have these conversations. They say, oh, wow, we have exa-cell. It's miraculous. It's going to change everything, except it's $2.1 million. And how are we going to afford that? And the real accessibility issue from the price. And that's a one-size-fits-all thing, right? It's the same drug you're giving over and over again to potentially hundreds of thousands of people if you could actually scale. It's a more complicated problem if you're talking about more bespoke, personalized treatments. But again, I think the same principle applies. If you can streamline the discovery process, streamline the regulatory process enough, I think we will get to the point where, it becomes almost like a CAR T, right? Like a CAR T therapy is a personalized therapy. You're taking someone's cells, you're processing them, and it takes some time, it takes some money, but you can make the drug and deploy. And now that's been deployed to hundreds of thousands of patients around the world. We're starting to see it with personalized cancer vaccines. So I think it's only a matter of time before we get gene editing drugs of various types, particularly base editing and prime editing, to that point.
I think it will be quite interesting to see what the interplay ultimately is going to be between the traditional development pathway of randomized controlled trials, etc., and investigator-initiated trials. Because the way that you're making it sound is the programmability of the editors is such that one can be very quick at introducing changes to the sequence, put it into the patient. And if you have an investigator-initiated trials, you can start treating patients that have specific mutations that you're capable of treating. Of course, that is not going to result in a regulatable product, but you'll be curing people. And at some point, there needs to be some change in the mindset in terms of how we currently view investigator-initiated trials, and instead of how we should be seeing them as true trials that can inform clinical development of products that can be then a bit regulated by the FDA and paid by payers, etc., etc. To me, that interface,
exactly.
I get the feeling that these advances are going to make it necessary to have this conversation about how we turn these investigator-initiated trials into more valuable pieces of the development pathway.
Yep, I think that's exactly right. I mean, you can envision a couple of different ways to try to go after it. You can envision it as more like these are N of 1 therapies in the clinical trials with your single patient in front of you. And so it's almost like single patient INDs. Yeah. The other way to look at it is you want to bundle all these single patients together in a larger sort of almost like a master protocol, like a more platform approach, and then be able to have the flexibility, right? So, I mean, I think there are a couple of different avenues to pursue with the regulatory agencies like the FDA. And, as I said, we're going to see some very interesting things happening in that space in the coming. We all see it coming. And, the FDA, they're very well aware that there's anything that lends itself to a platform approach, it's gene editing. And so they know the score, and they have signaled their openness to work with investigators like myself, certainly, to figure this out.
One thing I have heard, a kind of counter argument that I've heard, Kiran, is that if you're in a kind of therapeutic program, very often there's some engineering that has to be done with the editing system that you're using. This is the kind of difference between the kind of translational world and the world of research. In the world of research, you kind of, I'm going to do this. It's not necessarily optimized for my particular system, but obviously in the therapeutics context, the issue of I want my agent to give me the best possible outcome I can possibly get. So how do you deal with that?
No, it's a great question, because there is a tension between optimizing for every single scenario versus finding a adequate solution or set of solutions that broadly work across the patients you want to treat, right? Because it gets back into the questions of affordability and access. If you're really insistent on, I need to find the very best solution for every single patient who comes before me, that's a lot more challenging than finding, like a satisfactory solution. It may not be the best solution, but it will do the job. It will greatly improve that patient's quality of life, but it might not be the very best, most efficient, absolutely squeaky clean, safest therapy you can make. There's invariably a trade-off there, right? There's externalities that come into play.
I'm kind of really aware that I could talk about this all the day, Kiran. I find this so fascinating. But maybe we should kind of think about, like, in rounding things up, like moving on to this world of knock-ins and insertions.
Yeah, absolutely.
Do you want to talk a little bit about what excites you there?
Yeah, keeping my eye on the clock as well. So we talked about nuclease, we talked about base editing, and we've already exhausted the hour. But I do want to hit upon prime editing and its related technologies. And I also want to hit upon epigenome editing, because that was the other area where I think there was a lot of excitement. and a lot of interesting stuff that came out. So let me hit upon epigenome editing first quickly. So this is yet another form of editing. Does not make double-strand breaks. It doesn't even nick the DNA, but it's modifying the context of the DNA. It's not changing the DNA sequence, but it's modifying, making chemical modifications to the DNA bases. You're affecting the chromatin. More than that, you're actually focusing on the methylation status. And so what was really neat to see at this Keystone meeting was several talks, a couple from companies, Chroma Medicine, Tune Therapeutics, and one from the academic side, showing a lot of progress with epigenome editing. The two companies have been using CRISPR-based epigenome editing, and they showed off some great data, some non-human primate data, targeting the PCSK9 gene as it happens, and showing that in non-human primates, you can use lipid nanoparticles, you can deliver this CRISPR-based epigenome editor, you use a guide RNA or guide RNAs to target the PCSK9 gene promoter and put lots of methylation groups there, silence the gene, and seeing almost 100% silencing at the messenger RNA level because the methylation turns off gene expression, and seeing in mice and in monkeys persistent effects out to months or even as much as a year, which suggests that if you're trying to inactivate a gene, well, we know we can do that with nucleases. We know that we can do that with base editors, both proven out in human beings, and I don't think it'll be long before we see that starting to be done with epigenome editors. The flip side is that epigenome editors aren't going to be able to do much for you besides turn a gene down or potentially do the opposite, turn a gene up. So it really has to be tailored to your particular therapeutic application. The third talk that was not from a company, it was from an academic group, showed a clever way to not have to use CRISPR for epigenome editing, but going old school, actually turning the clock back maybe a couple of decades, using zinc fingers. And showing that with epigenome editing, it might actually be zinc fingers that may prove to be more useful because they're smaller, they're easier to pack into viral vectors. You can actually pack several of them into viral vectors, so you can tune several different genes at once. They actually seem to be better tolerated in cells. And so it's kind of funny to think, wow, we've come all this way, and yet we're going back to zinc fingers because that's actually might be what's going to work best for at least epigenome editing. So that's epigenome editing. It was nice to see the progress on various fronts there. And then prime editing, the most noteworthy talk was from Prime Medicine, where I got to say I was just so impressed because it was Andrew Anzalone, who's basically the guy who invented prime editing in David Liu's lab, and then went on to be a co-founder of Prime Medicine, and he was representing the company at the Keystone. And he gave an update on the preclinical progress of Prime Medicine's various programs. And I swear, he must have gone through a dozen of them, and they all look fantastic. Any presenter, doesn't matter if they're a company speaker, doesn't matter if it's me as an academic, we always put our data in the best possible light, sure. But still, it's very impressive to see, wow, this prime editing technology, they're just redeploying it in different variations across all these different disease states and really having impressive results, whether it's ex vivo, whether it's in vivo, whether you're using it to correct specific nucleotide variants in the same way or similar ways base editing can, or whether it's putting in small insertions or deletions, whether it's tackling trinucleotide repeat expansions. There were a couple of examples of that, like Huntington disease, Friedreich's ataxia, or, as you alluded to, doing more sophisticated things like doing gene insertion, like being able to do a multi-step prime editing or prime-like editing enabled process to actually be able to catalyze the insertion of large fragments or even full genes into the genome. And so it was actually a very impressive demonstration of all these different flavors of prime editing, all the different variations in one talk across a dozen different diseases. Now, we'll see how quickly any of those get to the clinic. Prime Medicine has guided that their first ex vivo application, they're hoping to take it to the clinic later this year. We'll see. But it's clearly headed in that direction. And even if it's not Prime Medicine, there'll be other companies that are sort of in the space and using prime-like technologies to do similar sorts of things. I mean, this is going to be an area to watch for sure because of the versatility of these technologies. The other thing I would add to this is the potential for metagenomic mining to find just really cool things that we just didn't even imagine were there. And one very impressive example of this came from Patrick Hsu from the ARC Institute. He presented something new that he had mined out of the genome, which he's calling Bridge RNAs. It's a totally different mechanism from RNAi, from CRISPR. It's RNA enabled, but it uses an entirely different set of proteins, an entirely different mechanism. And so these bridge RNAs basically come out of these mobile genetic elements, and it would take me too long to try to explain everything in gory detail, but he's released it as a preprint that was timed to his talk at the meeting, so it was a little bit buzzworthy because of that. But basically taking advantage of these mobile genetic elements that naturally exist and figuring out how they work, how they jump around, and discovering that there's this complex bridging RNA that basically is almost like bispecific. One portion of this bridge RNA specifies the target site in the genome, and the other part of this bridge RNA specifies the donor DNA. And so with this one complicated RNA bridging RNA molecule, it specifies both the donor and the acceptor or the target site in the genome. And what he was able to show is that, at least in bacteria, it's all in bacteria for now, right? So we'll see if it eventually makes the jump to mammalian cells, which would be very exciting. But he could show you can take one piece of DNA and stick it basically anywhere you want into the bacterial genome. You can actually engineer it so that the donor is one part of the genome and the acceptor site is another part of the genome. So you can catalyze inversions or do like complicated, funky stuff like that. And I'm watching this, I'm thinking, wow, I mean, there are some potential therapeutic applications if and when this actually gets to mammalian cells, because now, there are lots of diseases that are caused by chromosomal rearrangements, like hemophilia, for example, like something like, half the cases are due to one particular inversion on the X chromosome. It's very stereotyped because of homology between two different portions on the chromosome. And if you had a way to engineer just exactly the reverse flip, you could treat a lot of diseases that otherwise wouldn't necessarily be so easy to treat, right? So I think it just goes to show that there's a lot still waiting to be discovered. And so, we're thinking about nuclease and base and prime and epigenome, but, I could foresee in 10 years, like a dozen of different new things that we aren't even anticipating that's going to come out of metagenomic mining. And, some of them will be restricted to bacteria. They're not going to make the jump to mammalian cells, but some of them will make it to mammalian cells. And, in their own time, I think are going to have therapeutic potential.
Well, this has been very interesting here, and it's exactly what we were hoping to hear about. It's probably one of the most exciting areas in biology. As I told you in my email, the podcast is called The Mixer because we mix our interest in science with our interest in cocktails, but we know that you don't drink, so that's fine.
But maybe Kiran has a mocktail. Is it just plain old H2O, or is it some other concoction, Kiran?
Bloody Mary mix, perhaps.
There we go. Fantastic to hear from you, Kiran. It's been a while since we last saw another face-to-face. Hopefully, maybe I'll be down in UPenn one of these days. There's so much going on down there. It's an exciting time. It's an exciting place for sure. It must be an exciting time. Thank you so much for your time here and your insights. We really appreciate it.
Thank you so much, Andy. Thank you so much, Juan Carlos. It's been a real pleasure.
How about that, JC? So much going on in this field at the moment, I think. It's really interesting to see how far into the clinic the field has come since we first met, Kiran. What was it, over a decade ago at the Brigham?
Yeah, very striking to see the progress. It's been really inspiring, and I'm certain that the best is yet to come, to be honest. I'm sure there will be a lot of excitement in this area for years to come.
And what about the cocktail choice? Are you inspired by the Bloody Mary?
Yeah, I must confess that I'm not a fan of the Bloody Mary. No? I really am not. No. I've never made it, and I don't think I ever will. However, there's a rich bibliography about the Bloody Mary, and we're going to share with our listener a particular video that I find very informative. It's from a website called cocktailchemistrylab.com, run out of San Francisco by a guy called Nick Fisher. He shows you 3 different ways of making the Bloody Mary: the basic, the expert, and what he calls the chemist level. And if you can pull that one off, my hat's off to you, because it's a beautiful drink to make. So we'll share that with our listener.
Great. So everybody, enjoy the Bloody Mary, and thanks so much for listening. Until next time.
Cheers.
Cheers.